Hello, today's lecture is on fecal coliform. So we're kind of diving deep into a specific topic, in this case, fecal coliform, but I'm going to relate that to some of our local issues and then also relate that to the reading from Emma Norman, the eco-colonization piece, and think about how fecal coliform and its role in the environment is actually a form of eco-colonization that is impacting indigenous people in the Salish Sea. So why do we care about fecal coliform? I often offer people a glass of water and say it's full of fecal coliform. We care about fecal coliform and then ask if they want to drink it or not. But fecal coliform is an indicator bacteria. As the name suggests, fecal coliform is associated with fecal matter. And coliform is a type of bacteria. So these are bacteria associated with feces. And typically, if there's recent fecal contamination into our drinking water that we're swimming or playing in, we're typically not super excited about that. And as we'll learn, fecal coliform in and of itself will not necessarily make you sick, but it's an indicator of recent fecal contamination. So it's a proxy for fecal matter in the water or in the environment, something we typically try to avoid. And fecal coliform does have large impacts on shellfish fisheries, is one major concern. If there's a lot of fecal contamination in the water, we're not allowed to harvest shellfish. Again, for that concern of recent fecal contamination, could, along with that fecal matter, be some pathogens that can make people sick. I'll try to present a few examples of things getting better. And one of those is in Portage Bay, just north of Bellingham, just south of the border. Those waters have been closed, a section of those waters have been closed for 10 years due to fecal coliform issues. And then through some collaborative efforts, that contamination has been cleaned up by better sewage treatment, fixing leaky septics, better farm management, better runoff control. Fecal coliform has been reduced to the level that allows for shellfish harvesting. And that's kind of one of the things that I think is interesting about fecal coliform is that it's not a persistent pollutant. It's not like mercury in Bellingham Bay that's there and it's going to be there forever unless we figure out a way to physically remove it or cover it up. Fecal coliform is something that only exists in the environment for a short period of time, and so it does give you an option of proactive measures, corrective measures, and then seeing a response in the environment and being able to implement those changes and the changes in regulation. Before you go dig clams, one of the things you're supposed to do is look at the Washington State Department of Health's biotoxin map. And this shows a few things. One, it shows which beaches are open, which ones are closed, and why they are closed. And these closures can be due to biotoxins, which commonly are referred to as red tides. And so if there's a red tide, those phytoplankton make toxins that can cause human illness and even death. And so it's important to check that out before you go dig some clams. And then also there's contamination associated with fecal coliform or other sorts of runoff. And so the section that the article is talking about is this upper section of Portage Bay that had been closed, again, for about 10 years and now is currently open. So that's good news. That's positive news. That's opening up shellfish opportunities for recreational and tribal harvest. It's a symbol of the environment being cleaner and the shellfish which live in these waters being safe enough to eat. We've also seen fecal coliform illnesses not associated with shellfish, but associated in this case, it was an E. coli which is one of the Fecal Coliform species, and you might have heard of E. coli as it relates to an E. coli contamination on lettuce, or if you ate at Chipotle's, now you have E. coli, like that sort of thing. So it's for sure an issue on land. This is a case where some school children went on a field trip through some milk barns in Linden, and the barns hadn't been cleaned out well enough, that proper precautions were not taken, and cleanup wasn't taken, and the students got fecal coliform on their shoes and pants as they were walking through this barn, and some of them tracked it home and actually got members of their family ill, with 60 plus getting ill, 11 being hospitalized, and 6 having serious illnesses. And that's both the children themselves, and then if they track that fecal coliform home, issues with their grandparents or their younger siblings. And so it's a real issue that you think about in a few different contexts. So I told you that fecal coliform is associated with fecal matter, but I didn't tell you whose fecal matter. And so just kind of think to yourself, fecal coliform are found in the gut and feces of A, humans, B, mammals, C, birds, or D, all of the above. So pick your answer, hold it in your head. And fecal coliform are found in the gut and feces of all warm-blooded animals. It's not just mammals, not just humans, but birds can also have fecal coliform because they are warm-blooded. And so it's an interesting one, where it could be associated with like chickens or any other birds. And so it's an indicator of fecal matter. But seeing fecal coliform in the environment, it's hard to attribute that fecal coliform to humans or cows or dogs or pigs or birds. And so other work has to be done. Typically what's done is they do some DNA analysis, because that fecal matter will have DNA of the organism that released that fecal matter. There'll be some sloughing there, and there'll be some DNA there. So if you have so you're able to use DNA techniques to figure out what species left that fecal matter. But in general, we go out and measure fecal coliform, and we don't really know who it came from. We just know that it's there, and if it's there, then there's probably fecal matter, and we typically want to avoid that. Now, coliform bacteria is a wide group of coliform, of bacteria. There's total coliform and fecal coliform, and then E. coli. Now, you might have heard of E. coli, maybe you've heard of fecal coliform, and total coliform might not be something that you've been concerned with. But we can think of these either like in a bullseye model or sort of a nested model, where there's total coliform, and that includes environmental coliform. So coliform is a type of bacteria. So that includes coliform bacteria that can live in the environment, and it includes fecal coliform. And fecal coliform, as the name implies, are associated with fecal matter. They live in the gut and feces of warm-blooded organisms. They live in a very specific environment. Now, within fecal coliform are E. coli, and we hear reports of E. coli on lettuce or spinach or whatever it might be, and these big recalls of foods due to E. coli contamination. And that's due to concerns around human illness. Now, not all E. coli will make you sick. You can actually eat a whole bunch of E. coli and not get sick. However, if you eat E. coli 1057 H7, that will make you sick. So there's a specific strain of E. coli that can cause human illness. There's lots of E. coli. The majority do not cause human illness, but there's a few specific strains that can cause human illness. So again, when we detect E. coli, it's better to just avoid it and not roll the dice. However, the vast majority of E. coli will not make you sick. You have E. coli living in your gut right now, and only a specific strain of that can cause human illness. So you can think about it as E. coli that makes you sick is part of E. coli, part of fecal coliform, part of total coliform. Not all total coliform are E. coli, not all fecal coliform are E. coli, but all E. coli are fecal coliform and total coliform. So if that makes sense. So again, there's a wide variety of coliform, some of which are fecal, some of those are E. coli, and some of those are ones you don't want to get around. And we can quantify that a little bit more, where the total coliform are Enterobacter, Citrobacter, Escherichia, that's the E for E. coli. Escherichia, you can see why we just call it E instead of Escherichia, and Klebsiella. These are the 4 groups of total coliform. Of those, 2 groups are considered fecal coliform, the Escherichia and the Klebsiella. So the E. coli and Klebsiella. These are the fecal associated ones. So Citrobacter and Enterobacter are not fecal coliform, they are total coliform. You would be good to at least be able to match these or identify them as total coliform, and it would be worthwhile to be able to match these with fecal coliform. So fecal coliform is a group of bacteria that occur in the gut and feces of warm-blooded animals. They're facultative anaerobic, which means they can go both ways. They can use oxygen when it's present, and they can use anaerobic respiration when it's not. So they're happy if there's oxygen, and if there's no oxygen, they have a second, they have an anaerobic process as well, so they can use anaerobic respiration, which makes them really versatile. That makes them a really hardy bacteria that can live in environments with a lot of oxygen and ones with no oxygen. That's fairly, that's not very typical. Typically, bacteria are one or the other. They're rod-shaped in the gram-negative, red or pink. So in this staining, these are your fecal coliform, and these are other bacteria. So E. coli is a subset of coliform. It's common in the human intestine. So you right now have E. coli. You have E. coli right this second. It's not usually harmful. However, some strains can cause illness, and it's frequently used as an indicator of contamination. So the statement that's worth remembering is that if E. coli are present, there's a much greater risk of infectious microorganisms occurring, which can cause human illness. So again, we are using E. coli as a proxy for fecal contamination. And there's all sorts of things in fecal contamination that we don't want near us. And those things are many and varied and often hard to measure. But E. coli is nice and handy and is relatively easy to measure and count. So if we measure and count E. coli, which is a relatively straightforward thing to do, it gives us a good picture of what's in the water, and a good picture is if there's a possibility of fecal contamination in that water. And E. coli is this great indicator because it thrives in a specific and unique environmental conditions, which means that we can set up conditions in the lab where all the other bacteria will die, but E. coli will thrive. So you can think of it as like a sieve or some sorting mechanism where if you take a whole bunch of bacteria, we can run it through this process that sorts out all the other bacteria and leaves fecal coliform, E. coli in this case, behind until we can count the E. coli. One of their unique aspects that make them handy for quantifying is they grow rapidly at higher temperatures, 30 to 40 centigrade. Think about where E. coli live in your gut right now. Your gut is much warmer than most environmental conditions. If you think about Lake Whatcom or streams, they're typically much colder than your body. So those bacteria that are living in that environment are used to cool temperatures, and they typically are not able to thrive at higher temperatures. So in wintertime in particular, like local streams are down to about 10 degrees centigrade, maybe a bit less. Lake Whatcom, Puget Sound in the winter are kind of in that 12 range. So if you take bacteria that have been living at 12 degrees centigrade, heat them up to 30 to 40 degrees centigrade, they're going to die. Fecal coliform are going to be happy there. E. coli also ferments sugar or lactose to produce acid and gas. And so that's a food you can give them that produces an acid, and that acid can be used to do a color change assay. So you can measure pH basically by a solution that changes color as acidity goes up, as things get more acidic. And it also produces a gas, and so you can measure that gas production by trapping it in a jar and seeing how many bubbles it makes, more or less. And it also tolerates bile salts, and so because it lives in your intestines, it's able to deal with bile salts where many other bacteria would not. So you can think of the combination of all of these where it kills all the other bacteria with the exception of E. coli. So we can set this very specific environment where only E. coli is happy. There's a couple different ways of determining E. coli or counting E. coli. I'm going to talk about one that's the most widely used method that the state uses, that the Department of Health uses. And just because I think it's important to understand and visualize when we look at the data and we look at these numbers, what's behind them. And it's a pretty straightforward method, and it's a pretty visual method. So I think it's worth spending a little bit of time with. If you're interested in this more, you can take ESI 361, water quality, and we actually do this in class. So one of the primary methods is called the membrane filter technique. It's simple, fast-ish. You basically filter water, put it on a medium. Medium is another word for a food source, and you grow it up, and then you count the colonies, and that's your unit of measurement. So the membrane filter method, we typically take a 100 milliliters, what is that, 8 ounces maybe, of water, collect it in the environment, keep it cool, bring it back to the lab, pour it in a funnel, filter that 100 milliliters of water, and we'll learn later why 100 mils is our preferred number. Filter it over this filter here that has these nice little grids in it. And that's sucking all the water through, and all the water gets sucked through the filter, and the bacteria gets stuck to the filter. So what this is doing is it's taking 100 mils of water, separating the water from the bacteria, leaving all the bacteria behind on this nice gridded filter, which we'll see in a minute has a handy advantage. And then you put the food for the E. coli on this filter, grow it up, meaning incubate it in a little oven, basically, for 24 hours, and then you count up the bacteria. And you count the bacteria by counting these little colonies. It shows these little bumps, and I'll show you a picture of some real colonies here in a second. But the idea is that each individual bacteria, you can pretend one little E. coli lands right here in this little square. One singular bacterium lands right there. Over 24 hours, it turns into a little colony of bacteria, so a little bump, a little colony of bacteria. And we count these colonies, and we use those as a proxy for the individual bacteria. So our unit of measurement is colony forming units, CFU. So each of these colonies, we call a colony forming unit per 100 milliliters, and that's how much volume we filtered. So the unit is, if I take 100 milliliters, filter it, I count up the number of colonies 24 hours later, and that's my measurement. We can ignore the total coliform for right now. Here's an example of fecal coliform. So here's that gridded filter that was 100 milliliters of water was filtered through here. It was incubated for 24 hours at 44.5 degrees Celsius. After 24 hours, a number of colonies show up. 1, 2, 3, 4, 5, 6. We'll count them in a second. You count up these colonies, and that's the number of colony-forming units you have per 100 milliliters. And then second, we'll talk about like what that means in a regulatory standpoint. But I just want everybody to kind of have a good visual for what that looks like. The reason we do 44.5 degrees centigrade is every bacterium has its own temperature range. You could think about across the globe, from the poles to the equator, every organism has its own special range. If you take a bunch of penguins, bring them to the equator, they're not going to be happy. Same thing if you take a bunch of tropical animals, you bring them to Antarctica, they're also not going to be happy. The same idea with bacteria, that you have these cold, quote-unquote, cold-loving bacteria that really like it around 10 degrees Celsius. And that's kind of where we're at. Our freshwater temperatures and marine temperatures are about that in the wintertime. Your summer-loving bacteria might be up to 25 degrees centigrade, which we'll have in shallow ponds and shallow lakes, things like that. Your total coliform, we incubate between 35 and 37. Now that's giving you an overlap in your total coliform dashed line here, your total coliform bacteria and your fecal coliform bacteria. So you're allowed, it lets you grow both of them. Move me kind of in the way. Allows you to grow both your total coliform and your fecal coliform right here. The total coliform species start to crash and die out in the low 40s, where fecal coliform are happy over a pretty wide range, but hang on up until the upper 40s. And so if we push the temperatures to sort of the extreme end for fecal coliform, we've eliminated all of the other bacteria based on a temperature preference. So this is our plate again of all these different colony forming units. And we can count them up. Oh, I guess the next slide is going to count them up. I'm sorry. So our unit of measurement is colony forming units. The bacteria are quantified as colony forming units, typically reported as CFU, colony forming units, per 100 milliliters. One count per colony normalized by 100 milliliter filter volume. What that means is, let's say you accidentally filter 200 milliliters of water and you count 10 colonies. You would have 5 CFUs per 100 milliliters. You won't need to do this math in the quiz, but just as an example. Because you filtered twice the volume of 100 milliliters, so you would half the number of colonies you counted. Another way to think about it is if you filtered 50 mils of water and that resulted in 5 colonies, you would double that number to 10 colonies per 100 milliliters. So it's just a way to normalize it. It's not that important. But what each colony represents in this case, again, is that single bacteria where we're operating under the assumption that each of these colonies is the result of a single bacteria landing on our gridded filter, growing up into a little colony that we can count. In this case, there's 16 colonies on this filter. We filtered 100 milliliters, so we have 16 colony forming units per 100 milliliters. Now we do a 24-hour incubation because if you incubated for 2 or 3 days, these colonies would overgrow and just become these like massive super colonies, and it would be impossible to count the individual colonies. If we did 8 hours, it wouldn't be big enough. These colonies wouldn't be big enough to count. So 24 hours is our metric. And we'll talk about that kind of delay and what that means for regulation, but this is a biological measurement. We are using biology to tell us about the environment. This is not going out, putting a probe in the water, taking a measurement, writing it down right away. This is a multi-day process. So I told you a bit about fecal coliform, and we'll talk about fecal coliform and shellfish, but before we get there, for reference, how many fecal coliform bacteria do you think are allowed in our drinking water? 10 CFU per 100 mils, so that'd be 10 bacteria per 100 milliliters, which is 8 ounces-ish. I don't know off the top of my head. 100 bacteria 100 CFU per 100 mils, 1 CFU per 100 mils, or 0 CFUs per 100, 0 CFU per 100 mil. So think about it for a second. Lot, little, none. It's kind of, or in the middle. The regulatory standard for fecal coliform in drinking water is 0. You are not allowed to have any fecal coliform in your water. So we've decided the safe amount of fecal coliform to drink is 0. However, you can have much higher amounts of fecal coliform depending on your use. So drinking water, 0 CFU per 100 mils. For fresh water, it actually depends on what you're doing. Total body contact swimming will be 126 CFUs. And so if you're going to go to Lake Whatcom and swim around, the fecal coliform count has to be under 126 CFUs. If it's like a roadside pond where swimming is not allowed, but you can stand on a dock and go fishing, or you can just look at it, those values go up to be much, much greater. The idea is that with swimming, you're getting the water in your mouth and your eyes and your ears. There's a possibility of you consuming some of that water and some of those bacteria. For shellfish harvesting, you're only allowed to have 14 CFUs per 100 milliliters. So these are different standards for different types of uses. It'd be useful to know what the drinking water standard is, and the shellfish harvesting standard would be useful to know. Or if I gave you all 3 of these and like had you match to these, it'd be useful to know which one goes with which. So the National Shellfish Sanitation Program sets the national standards for fecal coliform and shellfish safety. And it's interesting, it's not based on the last sample that you took. It's based on an average of the 30 most recent samples. And so it's not just the last one you went out to collect, but it's the average of 30 samples. And this 90th percentile is a bit weird, but it comes to be important. The 90th percentile cannot be greater than 43 CFUs. What that means is 10% of your samples cannot be greater than 43 CFUs. You could have 100 samples, and 20 of those samples are 43. Do the math really quickly. Or 15 of those samples are 43. And if the other ones are 0, your average could still be less than 14 CFUs, but more than 10 of your samples, more than 10% of your samples are above 43 CFUs. So there's because the way that bacteria work in the environment, it's not a constant supply, particularly if the source of that bacteria is runoff. You can have a whole bunch of days that are 0, nice dry days, no runoff, and then it rains and all of this fecal matter comes washing out of the watershed and it's over your shellfish. You know, the average could be quite low because of all the dry days, but in that instant of having all this bacteria over the shellfish, it's not safe. And so we use 2 different measures: the average amount of bacteria over 30 samples, as well as the having 10% of them less than 43 CFUs. And we're going to spend a bit of time and talk about the Nooksack River, and this is a good example of pollution coming in from off the reservation. This is the reservation line. Pollution coming in off the reservation and closing down a shellfish fishery here on the Lummi Reservation. So it's really similar to that Emma Norman's piece about pollution coming in from outside of the Semiahmoo Reserve that's then impacting their ability to practice their inherent rights. That's extraterritorial pollution, meaning pollution that they did not generate within their borders, pollution that they're subject to that's impeding their inherent right to go harvest shellfish, which is a form of colonization, and Emma calls it eco-colonization, or it's called eco-colonization, which is an interesting way to think about how the degradation of our environment is a form of colonization, that if the environment is not healthy enough for you to practice your traditional practices, is that a forced method or forced mechanism for colonization? So we'll talk about one example here that's very local. We're just across the bay here at Western. Lummy's over here. The Nooksack River is coming down here. The Nooksack River drains Whatcom County. Whatcom County is frequently referred to as dairies and berries. We have a very strong agricultural industry here in Whatcom County. And we'll break down kind of some of the numbers of what that looks like. And it's not a bad thing. It's a great thing. It's a healthy economy, but we also need to balance that with all of the users of the space and ensure that the upstream impacts are not having downstream consequences. So when you manage shellfish, it's managed in discrete spatial locations. These different colored sort of blobs are the management units that Lummi Natural Resources uses to open and close shellfish areas. So they'll do a population assessment along, this is Lummi Shore Road, they'll do a population assessment along here and say, there's enough clams for people to dig here, there's not enough clams for people to dig here, here's okay, and here's okay. So that's all those are. In addition, there's all these little squares with numbers written on them. These are where the fecal coliform samples are taken. And we're going to look at some real data from the Washington State Department of Health in 1 second. And it's going to reference these numbers. Now, one thing of note is they're not totally sequential, like 49, 50, 51, 52, 53, 4, 5, I don't know where 6 is. Any of these other ones, 271, 272. So they're kind of all mixed up. But we can jump back and forth and do some camera 1, camera 2 if we need to. But just know that that's what these samples are. So our main thing of interest here are the squares and the numbers associated with them. Now here is a data sheet. This is an actual data sheet from the Department of Health for fecal coliform. Now we're going to walk through this pretty slowly and get an idea of what this actually looks like and what this means. So first we know this is the Shellfish Bacteriological Water Quality Survey. Our samplers are Bill and Curtis. So they went out. What day did they go out? They went out on May 6th, 2014 to Portage Bay. And they are doing an ambient monitoring program by the state. They are basing their tides off of Bellingham Bay's tidal charts with a high tide of 952 and a high tide at 5.6 feet and a low tide at 1704, which would be 5 o'clock basically, and a low of 1.3 feet. So they're giving the tide values here because this is an intertidal area, and at low tide you've got the fresh water running across the beach. So at low tide you're more likely to get more of the freshwater influence. At high tide the water's coming up, so if you're pushing some of that fresh water back in the Nooksack, and the shellfish beds are being inundated, covered in saltier water. So the value you're going to measure really depends on the tide, and so that's why they had to record that. We're going to walk across all of these columns of data here. Our first column is SAM. SAM in this case stands for sample. So Bill and Curtis went out and collected a whole bunch of samples on May 6, 2014. This specific page is dealing with samples 25 through 36. So that's just their internal. They start their day at 1. They end their day at wherever they end it. STA, short for station. These are the little squares I was talking about. So they started with 48 and went all the way around, ending at 58. Time, this is the time they collected these data. And tide is an ebb. Tides, we talk about, 2 common names are used. Flood, meaning the water is flooding in. It's now low tide, it's moving up to high tide. That's a flood tide. ebb tide is it's high tide and it's moving down to low tide. Our high tide was at roughly 10 o'clock in the morning. Our low tide's at roughly 5 p.m. They're going out around noonish, so the water is dropping. SWT. SWT stands for sample water temperature. When they took their 100 ml vial and scooped up some water, they also stuck a thermometer in the water to measure what the temperature of the water was. And on this date, the temperature was between 11 and 13 degrees Celsius. Again, Fecal Coliform are happy in that like mid 40s, so much cooler than that. SS, in this case, is sample salinity. Salinity is how we measure saltiness. Freshwater 0. Typical Puget Sound water is about 28. Full oceanic water might be 32. That's the amount of salt in the water. So 0, fresh, 28-ish, full marine water, kind of well-mixed water out in the San Juans, etc. Station description marks / BIDN. I don't know what BIDN is, but here's their notes. It's overcast. The river is muddy. Bunches of seals. And then way down here is a little asterisk. Our cow's gone. Okay. Overcast, that just gives you an idea what the weather was. The river muddy is an observation to notice that they can see fresh water there, and it's quite muddy. The Nooksack River is typically pretty turbid, meaning it's pretty muddy. Bunches of seals. Why would we care about bunches of seals? Out in this area, out kind of by Portage there's these little intertidal sand spits, gravel spits, that seals haul out on, and they'll berth there, and they tend to hang out there quite a bit. While they're hanging out there, they're also producing a fair amount of fecal matter. So noting that there's a bunch of wildlife in the area also tells you that, perhaps the fecal coliform is coming from there. Asterisk, are the cows gone? Portage Island has a herd of wild cows. That's the island off the Lummi Peninsula. And at low tide, you can walk back and forth between the Lummi Peninsula and the Portage Island. There's a herd of cows that got loose decades and decades and decades ago, and now it's this little self-sustaining population of wild cows. Cows produce a fair amount of fecal matter as well. Approximately 100 pounds of urine and feces a day per cow. So they are massive waste generators. Going into the lab ID, this is just our internal thing number to keep these things straight, and then dash 1 through 12. So that's just when the vial shows up in the lab so they can keep them straight. Before I get into these data, let's jump back over to these. So Bill and Curtis went out on May 6th. They put water in vials, measured the temperature and the salinity. They drove these samples in a styrofoam cooler to Fairhaven. They loaded them on a Greyhound bus. That bus went down to Olympia. In Olympia, they were taken off, and when somebody walked in the morning, they had a styrofoam cooler of samples that Bill and Curtis collected the day prior. When they got them, they recorded the date and the time. So it was the next day at 8:47 a.m. Somebody opened that cooler, and they had to check the temperature of the samples. If those samples were not on ice and they got up to 30 or 40 degrees centigrade, those fecal coliform bacteria would be just growing like crazy. So they want to ensure that the bacteria in that vial are roughly equal to the density of bacteria that they sampled. Meaning that if you took a sample of water, you left it in your warm car all day, those bacteria are just going to go crazy and grow and grow and grow and grow. So if you measure the number of bacteria in that sample, it would not match what's in the environment. So we check that. So our data is good, nice and happy samples. So now whoever's working in the ecology lab numbers the vials, they do the same thing we just talked about, filter it, throw it on a petri dish with some good food for the E. coli, let them grow up for 24 hours, and then count them. So again, we took our sample on the May 6th. The lab got it on the 7th. And then on the 8th, it's hard to read here, it says May 8th, 2014. So 2 full days later, they counted the number of colonies on that filter. So we start in our first one here, 49. These are CFUs per 100 milliliters. 49 is our first measurement. The 30 sample average for the National Shellfish Sanitation Program is 14. And you cannot have more than 10% of the samples above 43, I believe. So this is a number of concern. Move down one more. 350. 350. 350. 920. 540. 240. 49. So all of these are what you would say are bad numbers. This is saying less than 1.8, and so there's a little less than symbol here. This method doesn't let you get to 0. You cannot say there's 0 bacteria in the environment. You can just say that you didn't find any, and your method is only good down to 1.8. So that's as low as we're going to get. So that's a good one. That's a happy piece of data. That's not. 79, 540, 350. So all of these values are way, way, way above anything we want to be around. 920 is massive. So we can start to look at these data and think, well, that's interesting that some are near 0, some are 50, 49, others are near 1,000. That's huge. So what's going on here? Why is there that difference in just a small bay? Well, let's think about what could be the source of these E. coli. What could be the source of these fecal coliform bacteria? One source that's been pointed to is the Nooksack River. So the Nooksack River is draining a large area, a lot of agricultural area, a lot of hobby farms, a lot of people, dogs, a lot of septic systems that could be leaking. Lots of sources of fecal contamination that's going to come down, wash across the tidelands of the Lummi people, closing their shellfish harvest to no fault of their own, and with the question of what's their legal repercussion in this instance. But do we know that those bacteria are coming from the Nooksack River, or are they coming from those seals, or are they coming from those wild cows? Like, how do we get some inferences based on these data? And we can assume that if the bacteria values are the highest in the freshwater, and that freshwater is coming from the Nooksack River, and the bacteria values are lowest in the saltwater, then there's a connection between freshwater and bacteria. Whereas if it were the seals, it would be the highest in the saltwater. So let's look at our biggest value, 920, with a salinity of Z, of 0. Our lower values tend to be here, 49, salinity of 25, so nearly pure marine. 10 here, 20 is our lowest value we measured. 15s are relatively low. And the in-betweens, 6s and 10s, tend to be the in-between values. And so there's some indication here that when you move into the fresh water, you have more bacteria. When you go out in the further marine waters, you have less bacteria, creating the inference that the bacteria is coming from the fresh water. Now here are long-term data from 4 of those sampling locations we just looked at, showing what they've looked like over time. And this is pretty interesting. I'll move me again here. This is pretty interesting. So that whole area, that whole part of the reservation had been closed to shellfish harvest since really the mid-90s. And that was due to a lot of, in this case, it was due to a lot of bad farming practices. And EPA began enforcing it. The Washington created the Nutrient Management Act. Nutrient Management Act is a euphemism for Manure Management Act. Nutrients and manure are used interchangeably in the agricultural industry. So Nutrient Management Act is really Manure Management Act. which is it's a fair thing to say. So these were implemented shortly thereafter. They came up with a TMDL, which is an EPA guideline for total maximum daily load. This is like a cleanup plan that the EPA makes you create if your water quality is below standard. So they created one of these. They meeting their targets. They're working cooperatively to improve some of the industrial practices. In a decade from 96 to 2006 was a massive success story. That in 10 years and a whole bunch of work, water quality went from terrible to well below the threshold. Shellfish fisheries opened up. People are allowed to go out, practice their inherent rights, collect shellfish and sell them under a harvest permit, collect shellfish for personal use, for events and ceremonies. This area of the reservation has a massively productive shellfish fisheries. Clams love it there. They grow really, really fast. Great. Everything's great. Just digging clams like crazy. Everything's great. Things start creeping back up. They're still below the line. Still good. And then, boom, there's a closure here in 2014. A couple things led to this. One is this idea of, well, we don't need to put any more resources into it because it's been cleaned up. So the state and other agencies pulled staff that were doing enforcement and compliance work because the issue had been solved. So we don't need to direct resources towards it because everything is fine. It turns out over time things get a little more lax. Some things are happening maybe they shouldn't be. And that results in these values being above, this is that 90% line, being above that 90% line and creating a closure in Portage Bay. So this is an issue that we've seen before, 30 plus years ago, solved in the mid-2000s, back again less than a decade later. So these pollution restrictions does restrict shellfish harvesting in Portage Bay. And the Lummi Nation said this closure has affected thousands of families and cost millions in lost revenue. Now that's, and I want to disentangle a few different things. Lost revenue is important. Manila clams in particular can be commercially dug and sold. When those openings for digging Manila clams happen, it's usually right before Christmas. It's during the winter low tides. It's a ton of work. You have to go out in the middle of the night, 1, 2 o'clock in the morning, lug a bunch of buckets across the mud flats, dig a bunch of clams, haul these hundreds of pounds of clams back up. You get like $1.60 a pound. So if you dig 200 pounds, you get like $300. That's a obscene number too. 200 pounds of clams is a lot. But anybody can do it. You just need a bucket and a shovel or a rake. And it happens right before Christmas. And so it's one of these things where a lot of parents will go out and do to get a couple $100 extra right around Christmas, give presents to the kids and things like that. And so it's, so like the money is important, but it's like when the money comes is also really, really important. And that amount per person isn't that much, but it's a really vital piece of it. To me, the bigger piece is also the impact on the inherent ability to practice inherent rights, the ability to even go out and shellfish, the ability for grandparents to take their grandkids out and dig clams during the summer on a nice warm day. That to me is a much, much larger infraction. But they're faced again with this issue of this extraterritorial pollution coming in from outside, impacting their resource. What do you do? In this case, really led by Tim Blue Jr. and others, the Lummies and dairy farmers came up with a cooperative agreement called the Portage Bay Agreement, where in this case there's some compensation from a few dairies. There's about 6 of them signed on. And they said, we'll offset some of the cost of that. We'll implement best management practices and exceed the state recommendations. We'll do the best we can do to ensure that we're not polluting the water. We're going to encourage other dairy farms to do this as well. And in this exchange, we're also going to be exempt from future litigation. So Lummies and the dairy farmers, a subset of dairy farmers, about 6 to 8, got together, came up with this agreement, and said, okay, and now we have this agreement. We're going to offset some of the costs from these closures. We're going to do the best we can to make sure we're not contributing to it, and we're going to solicit other dairy farmers to come and join. I think it's a really good example of cooperative agreement. If it ended in litigation, neither party wins. It's going to cost way, way, way more. It's going to take years of which are going to be lost and can never be recouped. So this cooperative agreement was entered in about 2017. We'll get to kind of how it's doing now. Chairman at the time, Tim Blue Jr., said, unfortunately, for the second time, our tribe did have to close our shellfish beds in Portage Bay. The Lummies have been harvesting shellfish here for thousands of years. It's who we are, he says, enshrined in the 1855 Treaty of Point Elliott, the same treaty giving tribes half the salmon in the Supreme Court. If we can't practice the harvest, our way of life is compromised, and so is the treaty. So also what he's referencing there is this impact is an impact on the treaty right. As such, the federal government can get involved, and it's a local county policies. Local counties are then impeding a federal treaty, and the federal treaty supersedes any local policies. And so, in the end, that's kind of the end card they can play in federal court and in litigation, but that's going to take a lot of money, take a lot of time, create a lot of bad will. In the cooperative agreement, the way that they approached it is the dairy farmers and the shellfish diggers, the Lummies, said, really, we want the same thing. We want a healthy environment, we want clean waters, and we want our grandchildren to inherit what we do. And on our current path, that's not likely possible. So can we come together and figure out how we can create a path for both of our families to be able to have our grandkids do what we're doing now? And that was kind of the foundation of coming together around the Portage Bay Agreement. I think it's a really cool example. I'll post a link to the Portage Bay Partnership, and you can actually read the agreement there. So we've been talking about dairies, and I don't necessarily want to make them out to be the sole source of fecal coliform, because they're not the sole source. But when you think of the sheer magnitude of it, they're a big source. Properly managed will have very little impact on the water quality, but a bad actor can have a massive impact on water quality. So in the Nooksack watershed, and these are all, I've actually used all conservative numbers in this because the numbers are just so crazy. In the Nooksack watershed, there's 87,000 plus cows on county farms, in Whatcom County farms. So not all those are in the Nooksack watershed, but there's a lot of cows in the county. 87,000 plus in Whatcom County. Not all of those are in Nooksack. Each cow produces about 82 pounds of manure per day. That's a conservative number. 100 pounds is frequently used, but I want to use a conservative number for this. That equals about 6,000 pounds of manure waste each day. So right now, today, in Whatcom County, there's 6 million pounds of manure produced. 6 million pounds of manure produced today. And to get that into human values, so it's a bit different, and not to gross you out, but our waste, human waste, is more nutrient dense than cow manure. Cows eat a bunch of grass, a lot of it's not digested, and it passes through. Humans tend to eat a more nutrient dense things, processed grains, meat, cheese, even a vegetarian diet. We extract more of those nutrients out, and we pass more dense fecal matter that has higher values of things like nitrogen in it compared to cow manure. So you can't do like a simple pounds per pounds, but in nitrogen space, which is a major nutrient and a major nutrient for plant and phytoplankton growth, and is a nutrient that we have too much of and causes eutrophication, overabundance of phytoplankton that then results in low oxygen and a lot of environmental issues. Having 87,000 cows in Whatcom County releasing 6 million pounds of manure per day in nitrogen space is the equivalent of having about 1.2 to 2.5 million people practicing what's called open defecation in Whatcom County. So it'd be like 1.2 to 2.5 million people just doing their business in the bushes in Whatcom County. It's a fair number. It's a lot of nutrients being released in the environment. And there's a couple farms that have gotten in trouble and received some fines, $7,500 after like a number of infractions. And you can follow here and see some what the Whatcom County agriculture looks like. But I think it's interesting, this partnership, and it actually is working. Let's go to the next two. This is a quote from Richard Pell from Pell Farms in Ferndale. They make not Gouda. What is it? They make the Gouda. Yeah, they make a bunch of Gouda and different cheeses. But he's saying we want the same things as Lummi want. We want to have a future, and we want to have that same future for our kids. And so we want to join this and work together. Here's a link to the agreement. It's Whatcom Family Farmers. There's also a Portage Bay agreement website. There's not a whole lot on there. The goal is to reopen the shellfish beds, which could take at least 2 or 3 years, if not more. It does take time. This is when Tim Blue was chairman. And then recently, in April 2019, for the first time in years, members of the Lummi Nation are able to harvest shellfish from 800 acres in the Portage Bay during the spring starting this month because of improvements of water quality. So this is what's called it's a conditional opening, or it's only open during certain periods of time through April 1 or through June 30. So still not open year round. But this agreement has implemented practices, drawn some light to the issue, and helped get the shellfish beds open at least through April through June, and more work is continuing. And so it's an important way of approaching this through a cooperative lens. I think that model has a lot of utility. It's, of course, cooperation where one side, Lummi, has a pretty good, their backup plan is litigation, which they would likely be successful in, but it would likely take a long time, and it would likely cause a lot of ill will. And it might result in the same thing, but much, much further down the road. And here is helping, I think it's helping bridge this divide between the agriculture industry and the indigenous communities in the area. That many things that they disagree upon, but there are some common grounds, and really focusing on that's important. So I really like this case. It's very local. I think looking at Emma's paper is also really important because that both has this eco-colonization built in, but it also has the difference of treaty rights between a Canadian First Nation and a U.S. treaty tribe, even though those two tribes were very much in overlapping territory, the Semiahmoo and Lummi, based on where they wound up being placed on this 49th parallel, are now living vastly different experiences. I think that's a really, really cool piece, and I think reading that will help you out quite a bit.